begin. Welcome everyone to our Centers of Excellence in Hematology Meet the Expert seminar series. Today we're pleased to have Brian Hayes from the Fred Hutch speaking to us today. Brian runs the Canine Core facility at the Hutch that studies hematopoiesis and transplantation. We'll have him speak with us in just a minute. I first want to just share my screen for a minute. Let me get there. That's fine. And then I will get to, now you can all see yourselves, but I'm really going to get to what I want to show you, which is somewhere here. Sorry. Come on, let me get it. Okay. We'll get there. Apologize for that. Okay, now we'll get to sharing the screen. I just want to show you the website to just make you familiar with our website for the Centers of Excellence. Can you guys see that now?
Yes.
This is the website. It's very simple to get to. It's cceh.io. And if you go on there, you can find lots of announcements here about our Meet the Expert series, about a monthly microscopy series that we're sponsoring. If you're interested in microscopy and are interested in using those tools, I highly recommend hopping on this seminar series. I've done microscopy for probably 40 years, and I've learned a lot of things from this series of the importance of choosing the right tools for your analysis. Further down the line, you'll see these Meet the Expert seminar series. And if you click on these, what you'll find is the series for this year. And each of these is made available as a recording. And so you can click on these links and hear these talks at later times. You can go review the talk if you want to. If you haven't had an opportunity to listen in, they are made available there from this last year and previous years. The other thing I want to show you is if you're looking to try and find services that are made available through the Centers of Excellence in Hematology, you can click on this service link. And what you will find here is all services. You can click on that. Maybe you have a question, how do I get my cells of interest? If you click that link, what you'll find are different centers. For example, this GMP cell processing center, which is made available. You can get you the cells of interest from, get help with that from Cincinnati or Yale, who can help you find cells of interest that you might be interested in studying. Furthermore, you can also, if you want human cell lines, you click on this. CD34 cells are available from the Fred Hutch and also from Yale. And if you click, for example, on the Fred Hutch's Hematopoietic procurement site, you can come here and find the services that are available, who to contact, and how to get help finding the cells that you are interested in utilizing for your research. So please visit our website. You can find a lot of core services that are available across the country. You don't have to be a local individual for those tools. And today we have the pleasure of hearing from Dr. Brian Hayes to tell us about the core facilities available at the Hutch that utilize canine models for understanding hematopoiesis. And with that, I'm going to just invite you to mute yourselves if you're not speaking, but Dr. Hayes has agreed to allow you can either populate questions in the chat and I can ask them, or you can unmute yourself and ask them throughout the presentation. So thanks for attending, and Dr. Hayes, your turn.
All right. Thanks, Diane. Let me just get this. Okay. Is this on the slideshow?
It isn't. It is not yet.
Okay. Let me have to change the monitor. I'm sorry about this.
Not a problem. It could be me messing you up there.
Maybe I'll try this. Does this show the slideshow?
It is not still. It says multiple participants can share simultaneously. So I think I've selected what you need. I'm not sure why it's not
Let's try
coming up.
this.
There we go. Now it's starting. Now we're up.
Okay, sorry about that. I have multiple monitors, and apparently I'm not too good at deciding which one is which. All right, well, thank you, Diane. So what I wanted to talk to you today about is the FHCRC CCEH Canine Core. And I just kind of wanted to give you an idea of what it is we do, what we can do, and see if anybody has interest in incorporating the dog model in their current research. But before I get to this description, I figured I would address a question that some of you might have, which is why dogs? Why not some other animal model? And the answer to this it goes back to the development of bone marrow transplantation and research that has been occurring at Fred Hutchinson since its founding. So conveniently enough, being at Fred Hutchinson, I was able to just go down the hall and get a firsthand account of the development of bone marrow transplantation and the use of the dog model in perfecting that technique from Reiner Storb, one of the giants in the transplantation field. He's been involved pretty much since the beginning. So the next couple of slides are from information I gleaned from conversations with Reiner and a couple of adaptations from slides he actually provided for me. So I appreciate Reiner taking the time out of his day to chat with me. So I'm going to start this story in the mid 1950s, and that's when a couple of researchers reported that after giving mice a lethal dose of irradiation, they were able to save that mouse from death by transplanting bone marrow from another individual. And this was really exciting news for a lot of people, because it was known at this time that after people are exposed to high doses of radiation, that pretty much that obliterates the blood producing cells, and people die shortly thereafter of lack of blood cells. So in the mid 1950s, there was a Cold War going on, and both sides in that Cold War were really building up their arsenals of nuclear weapons. So it wasn't beyond the realm of possibility that people could be exposed to high doses of radiation. And a lot of people were interested in having some sort of fallback plan to treat that anybody exposed in case that happened. There was also a group of physicians that thought they could use this technique in an actual therapeutic manner. So people with hematologic diseases could be irradiated with doses of radiation that will eliminate the abnormal bone marrow, and then transplanted with bone marrow from another individual, thus curing the disease. So people were excited in the mid 1950s, and they started transplanting humans with bone marrow from other individuals. And you can see from this graph, this is from a paper published collecting all of these reports. It really started to take off, this human bone marrow transplantation, up until about 1962, and then kind of dropped off to pretty much nothing. And this paper reported that of the 203 patients that were reported in the literature at the time of this publication, 200 of these patients had died. So really, the survival was abysmal, and it really looked like this was not going to be an effective treatment for hematologic disease. And by the mid 1960s, most people thought that these barriers to transplantation were insurmountable and they couldn't really be solved. So that's kind of the situation where we're at the mid 1960s.
Ryan, can I interrupt for just 1 second?
Yeah.
I'm not sure, your microphone is picking up some background sort of clunking.
I
that's
know it's,
happening. And I don't know if that's your chair or what it is, but I'm wondering if there's
it,
something we can do about that.
it could be. I'll hold the microphone away from my shirt as well. Maybe it's rubbing. So
Thanks.
so pretty much some of these hurdles to transplantation, why were the transplantations failing in humans? And the majority were due to graft rejection and graft versus host disease. This was about 175 of those 200 patients. And then there were some infections that occurred and recurrence of underlying disease that also led to mortality. So the mouse model was not very predictive of the outcomes in people. And a lot of this was due to its small size and inbred populations. And even in attempts to cause graft versus host disease in the mouse, it was very mild and they could recover from this. So even though a lot of investigators were abandoning the field of bone marrow transplantation, there was a small group in Seattle that was confident that this could work. So in Seattle, there was the lab of Don Thomas, and he had Bob Epstein and a brand new fellow in 1965, Reiner Storb, and they decided to use the dog model to try to address some of these complications of human bone marrow transplantation. Don had been working with the dog model for about 10 years at this point, and I really thought that that was going to be effective to address some of the issues that they were seeing with human mortality. So over the course of about the next 5 years, they put out an amazing amount of work and really started to knock down some of these obstacles to human bone marrow transplantation. They developed methods to store bone marrow so that you didn't have to have fresh marrow from a donor. You could actually freeze it and then transplant it at a later time. Figured out some of the cell doses that were required to get engraftment. Conditioning regimens were developed for whole body irradiation and cyclophosphamide treatment to eliminate the recipient's bone marrow prior to transplant. Some advances were made in histocompatibility matching. It was kind of rough at this time, but they used the dog model to kind of figure out how to match litter mates at this time and get transplants that would actually take. And then there were also some big gains in controlling graft versus host disease, particularly with methotrexate. So here's an image of a couple of the successful transplants. All these dogs are multiple years after transplantation, and they're 100% donor. So they were transplanted from another individual, and their entire blood system is comprised of the donor's blood. So most of these guys had total body irradiation, but this tall fellow in the middle had cyclophosphamide treatment. But this is an indication that allogeneic transplantation could actually be achieved in the dog model. And then these gains that were made were actually directly transferable to human patients. So within a short time, Fred Hutchinson Cancer Research Center opened its doors, and bone marrow transplantation became a critical component of the Fred Hutch clinical care from the beginning. And it wasn't perfect at this time, but the Canine served to help refine some of these bone marrow protocols. And I just want to pause for a second and remind you that this is only 10 years after a lot of the field had thought that this would never be accomplished. So it's really a short amount of time that passed from pretty much 100% failure to a really clinically viable treatment for hematologic diseases. So because of all of this work and advances that were made in bone marrow transplantation, Don Thomas was awarded the Nobel Prize in 1990. Since it's Nobel season, I figured I would show you guys the actual Nobel that he got, which is really cool to see. You walk in the entrance to Fred Hutch. But this prize was due, at least in part, to some work that was done in the dog model. So it's kind of nice to reflect upon the history that led to the awarding of this prize and the dog model's use in getting to there. So for transplantation, some of the answers to why use dogs is that they're large size and similar immune response to humans. Their relatively outbred status and large litters allowed experiments to be designed to overcome those hurdles that I mentioned were really preventing successful bone marrow transplantation in the mid 60s, and those being largely graft rejection and graft versus host disease. So that was just definitely an example of how beneficial dogs can be to gain knowledge on clinically relevant strategies. So in the intervening decades, the dog has continued to direct new transplantation protocols. This is not an exhaustive list, but just a couple of high points that I wanted to bring your notice to. One of them is non-myeloablative transplant, which really expands the population that can receive bone marrow transplants. The conditioning for myeloablative transplants can be pretty rough, and some individuals aren't quite healthy enough to receive that sort of treatment. So the non-myeloablative treatment is a lot more gentle, and that expands the population to some older individuals who particularly have a lot of hematologic diseases. So that was really a big advance that was facilitated by research in the dog. And another kind of in the same vein protocol that has been worked out in dogs is radioimmunotherapy. So there is an isotope in this situation bound to a CD45 antibody. So this radioactive isotope targets the recipient's immune cells, all of the hematopoietic cells, and then specifically eliminates them with minimal damage to surrounding tissue. So this is, again, a more gentle conditioning regime that was worked out in the dogs and then translated to human patients. So that's kind of a brief summary of kind of how we got to where we are today. And now let's talk about why we think the canine core is useful to the broader hematology community. And it's because of these decades of work at Fred Hutch in transplantation that we have actually a pretty sophisticated housing and treatment facility, and the staff with the experience to do a lot of these specific hematology protocols, such as transplantation, radiation, apheresis. And both of these things, the facilities and the staffing, can be obstacles to someone who might want to start a dog experiment on their own. But we feel that we can offer up our facilities and our experience to someone who wants to design an experiment remotely, and then they can make use of this model if they do so desire. So the two main arms of what we do here in the Canine Corps is specific canine molecular biology services, mostly related to transplantation. These are MHC typing and chimerism analysis. But we also have in vivo experiments that we make available to researchers remotely. So the details of this isn't really important, but this is kind of just a workflow of just typical MHC typing that we do for the dogs. And all of these steps individually are pretty simple, but kind of stringing them together. And if experimenters don't necessarily have this on hand, or sometimes clinician veterinarians need this information, they can't really do it. So we feel that we can save people some time and some effort just by offering up this as a service. So what we do is we sequence the variable regions of DLA88, DRB1, DQA1, DQB1. These 4 genes are pretty indicative of if they are matched, it will be a successful transplant. And if there's mismatches in some of these, we really would recommend not performing a transplant in these cases. One advantage of having us do the typing instead of people doing it themselves, sometimes there are weird events that happen. The canine genome is a little strange sometimes. And here's just an example of one of the oddities that we see. I saw in one individual that I was typing, and this is in the DQB1 gene, the variable region from an individual. And we only expect 2 alleles, one from the mother and one from the father. But what we see here is this location, we see a signal from 3 different nucleotides at this position. A G, a C, and a T. And a little bit less obvious, but also at this location, we see a signal from a G and an A and T are pretty much overlapping. And what we found when we cloned this out was there were indeed 3 separate alleles. And I interpret that as just that there's a tendency in this location to have duplication events in the dog, which will result in 3 alleles occasionally showing up in. I've seen them in DQB1, DRB1, and DLA88. So there was a paper published about DLA88 and used some long-range PCR, and this paper suggested that yes, there was a duplication event, and the DLA88 kind of replaced another gene that was that is a class 1 gene, DLA12, and they definitely saw 3 specific DLA88 genes and expression of these DLA88 genes. So these confusing events can be a little challenging if you're doing this sort of molecular analysis in a different manner. So we're happy to help people out when we can. So another molecular analysis that we do is variable nucleotide tandem repeat analysis. So these regions of the genome will be different frequently between 2 different individuals, just a different number of repeats in between 2 primers. So we have a couple of locations that we usually look at, and when we can tell these individuals apart, we use it as an analysis of donor and recipient contribution after transplantation. So we can see the percentage of blood cells that come from the donor and the recipient based on the different sizes of the bands that are amplified in these regions. The problem with some of these is that it's not a very sensitive assay, and sometimes it's hard to find bands that are differentially sized between individuals. So because of this, recently in June, I was approached by an investigator at Ohio State University, Bill Kisseberth. He's interested in tracking cells that he had transplanted in an adoptive transfer protocol, tracking them in the blood throughout their lifespan. So these cells were, in this case, NK cells, and they're only going to be transiently present in the peripheral blood. It was actually transplanting pretty small numbers of cells. So we wanted to develop a more sensitive method of detecting these cells than the VNTR, but also we didn't want to make it specific for Bill. We kind of wanted to make something that's more of an off-the-shelf reagent that we could use for any other investigators that kind of want to use adoptive transfer and track cells that they may be small in number and disappear over time in the blood. So Bill asked a colleague of his, Carlos Alvarez, who has a lot of, who's also at Ohio State University, has a lot of experience with canine genomes. And Carlos found about 10 regions with SNPs that occurred at about 50-50 within dogs, kind of regardless of breed. And 8 of these were pretty easy to design primers to that I had found, and design probes that would be specific to one SNP or the other. And what I'm showing you here is that here's an example of 2 siblings from our Fred Hutch colony that I just took blood from, and was able to see that pretty much they were identical at 7 of these 8 locations, but indeed they were different on chromosome 9 at the SNP location. So I think that this will have some applicability as an off-the-shelf product to just tell any 2 individuals apart, and we can use these primer probe combinations in real-time PCR to detect small numbers of cells within a larger population. So we're kind of excited about that. We still got some work to do on perfecting this this strategy, but we're pretty optimistic that we should have an assay that's developed pretty soon.
So Brian, I
Yeah.
want to ask a question in there. You're taking, so a transfer is done, you sample blood, and then you extract out of that blood sample to try and find these alleles. Is that right?
Yeah, so typically what we would do is collect some peripheral blood, and then we would see if we can detect a signal out of the DNA of these SNPs. So
So just as a, sorry, I'm just trying to refine this and understand it. Maybe, I don't know if other people who are listening have this similar question. So they want to follow NK cells. Are there flow antibodies for canine cells? How are they isolating the NK cells?
There are some flow antibodies. I actually don't know what Bill's isolation technique is. I think he might be culturing them, but the problem with some of the flow is that it might not be sensitive enough either. So on the lower level of the transplants, that the adoptive transfer that Bill's performing, there are like 1 million cells per kilogram. So the volume of canine blood, depending on the size of the dog, could probably be around a liter or so. And if we're sampling such a small volume of it with a couple mils, and there's only a couple million cells floating around in the blood potentially,
Sorry.
flow cytometry might not be sensitive enough. The PCR detection should be much more sensitive because you can pretty much detect, single copy theoretically.
Sure, I appreciate that. I was just trying to figure out how they were getting the cells originally to be able to do the transfer and then follow those cells. I get the PCR piece that you have designed. I just didn't get how they were following the cells to purify them to
Right,
for the
right.
transfer.
Yeah, and I don't know 100% the details on Bill's end on how he purifies the cells. But
Okay, that's okay. Thanks.
so apart from the canine specific molecular assays that we also offer canine core in vivo services. And this pretty much just is composed of drafting IACUC protocols, coordinating with our comparative medicine staff, and collecting and processing samples if necessary, freezing them back, and then updating the investigator on the progress of these experiments and delivering results. So I'll just walk through a couple of examples of some of these in vivo experiments that are currently ongoing. So this experiment here, this was actually published just recently in June in Blood Advances, and it's an experiment that Carol Mao at Seattle Children's initiated, and she's interested in treating hemophilia with by transfecting plasmids into the liver. So what they describe in this paper, it's really kind of interesting. They thread in a catheter and guide it with fluoroscopy into the liver, deliver an encapsulated plasmid, and then use a transcutaneous ultrasound. So an ultrasound producing device on the skin to disrupt this encapsulation on the plasmid and encourage the hepatocytes to take in the plasmid. And then this paper shows that they had sustained expression of factor VIII from plasmids that they transfected into a couple of dogs that they did at our facility here. So I wasn't specifically involved in this study, but currently we're doing studies that work off of this experiment, and we're trying to modify the strategy to see if we can figure out maybe a little bit less invasive approach. So this is really kind of exciting work. Carol is really gung-ho about continuing this in the dog. So why would we use the dog for this experiment? A large part of it is the scale. Mice are, a lot of the preliminary work was done in mice, but the lice are too small to actually like adequately represent like what the situation might be in humans. You don't have to thread in catheters to mice to deliver plasmids to the liver. But the size of the transcutaneous device is also important in this feature. One additional benefit of the dog is that we can draw blood repeatedly to track the expression of the transgene in the peripheral blood. So regular blood draws, daily will give an idea of what the expression is of, in this case, human factor VIII. So eventually, one more advantage of this dog model is that they have a hemophilia dog model in North Carolina. So Carol is planning to translate her experience here in dogs to try to treat those hemophilia dogs. And like I said, eventually work on translating this work into treatment for humans. So it's an exciting area of work that we're proud to be a part of. So another example of a current in vivo experiment we have is one that's initiated by Reiner Storb. And he wants to determine whether mobilization of stem cells is going to affect the exposure and the elimination of 117 c-KIT positive cells when they're exposed to a radioisotope. So CD117, like other animals, is expressed on canine hematopoietic stem cells. And the idea is to have an antibody against CD117 that is bound to astatine 211, which has a short half-life of 7.2 hours, decays by emitting an alpha particle. It's got a short half-life, so it shouldn't damage surrounding tissue too much. And we're really uncertain whether or not mobilization, in this case with plerixafor, is going to change the exposure of CD117 to the antibody and allow more of the cells to be eliminated. It's conceivable that it's such a large volume of bone marrow that some of the cells could just kind of remain in their niche and be protected from antibodies. So the thought is that mobilizing could really help eliminate either the normal cells in terms of for transplantation, or if in terms of leukemias expressing CD117 to expose more of those cells to this antibody that will eliminate them. So in this case, why use the dog? Scale is another part of this benefit of using the dog model in this case, and history of translating the dog results to humans. So pretty confident that results from the dog in this experiment in particular will be reflective of results we would expect to get in the human.
Brian, I have another question. Sorry.
Yeah.
You know, I'm thinking about animal numbers. In a canine, how much variability is there to get statistical significance for your results if you were going to use a canine model?
So, I mean, that can be an issue because you don't get large numbers like you do in a mouse. I mean, we're not necessarily limited on the number of animals that we can do, as long as they're
Yeah, no, I'm wondering about the variability in whatever the experiment is within that canine model, to know, to help determine, you know, a power calculation for how many animals you would have to do.
Right. Typically, a lot of the experiments that are done, they have pretty clear results, so they're not slight differences from what we have been. But in this instance, specifically, this experiment is kind of like a trial experiment, and we'll see if there's a noticeable difference between the two conditions, mobilizing or not. And then if it looks promising, we'll move on to incorporating more dogs in the study. So this one's actually a small scale. There's only 2 animals in each
I
arm.
see. Okay.
But yeah, the plan is, you know, if successful, that we'll move on to incorporating more dogs into the study.
Okay, thanks.
No problem. So that's just an example of a couple of in vivo experiments that we've got going on, but I just did want to hit on the fact that we do have 2 germline mutations that model hematologic diseases, pyruvate kinase deficiency and X-linked SCID in the colony. So if someone is interested in these 2 diseases, these dogs are available. So just a little explanation, the pyruvate kinase deficiency is the result of a point mutation in PKLR, and this leads to ineffective ATP production, which results in a couple of red blood cell related morbidities, but most strikingly it's anemia. The affected animals usually have a tough time, you know, running around and doing all the things that they want to do. They have to they have to rest a little bit more than a regular dog. And we don't currently have any experiments going on with these animals, but they are available if people are interested in using them for experiments of pyruvic kinase deficiency. So the other mutation that we have in the colony is an X-linked SCID mutation, and this is a 4 base pair deletion in IL-2 receptor gamma, leads to defects in B cell, T cell, and NK development. These animals are extremely immunodeficient, and they do need to be treated within 3 to 4 months of birth. But definitely recently, Hans-Peter Keen has used these animals for some gene therapy experiments, and he has treated some of these dogs with IL-2 receptor gamma transduction in stem cells, and had some successful results for that. So currently we're not doing any experiments with these dogs again, but they are available if people have a desire to look into this excellent SCID. So pretty much, I think what I've tried to represent today is some of the reasons why you would want to use dogs to perhaps not completely replace other animal experiments, but at least to supplement some of the information you're getting from some of your other experiments. Here I just have listed, some of the attributes of a dog that kind of differ from a mouse. And one of the key features that they've been used for at the Hutch is directly transferring protocols that are successful in dog to humans. They've really had a long history of predicting response in humans. So that's kind of one of the things that they really are a good model for. Some of the other things that I didn't quite hit on is that they're long-lived. If you're looking at if you're looking to track something for multiple years, mice aren't necessarily the best option for that, but dogs will be able to provide answers, in a situation like that. So don't know what the time is, 1:45, which actually is pretty good. If anyone's interested in incorporating some canine work into their own experiments, can email me directly, and we can talk about some of the positives and negatives of what we have available. And with that, I'd just like to thank everybody who's involved in this. And if anybody has any other questions, I'm ready.
Okay, thank you, Brian, for that presentation. I still, we get so narrow, you almost never think about like going outside your mouse or cell culture model or something like that. And I'm really trying to think about how you could utilize the canine models. Do you guys do, for example, if you had a hemophilia or you had some other anemia in the dog, do you do drug screens in dogs?
So it's kind of convenient that the dogs are, pets and are treated a lot by veterinarians. So there are definitely a lot of standard clinical sort of tests that can be used. Hemophilia, we don't actually have those dogs specifically here. So I'm not 100% sure on what kind of clinical tests they have for that. Typically though, yeah, we can get standard blood counts, chemistries for clinical results that you need. So you can track those things pretty easily in the dog.
Okay. Other questions that anybody wants to ask?
I would just kind of, and thinking about, it seems like for any other type of work, you have to have a large amount of preliminary data to justify something like this. And, it's not like anybody's got all this preliminary data in a canine. So how can you justify, a transition into that type of model?
Well, so a lot of times, or these days, people kind of take some of the results they have in mouse work, and then they'll use the canine as kind of like a next step in a preclinical model. That's kind of like what Carol has done. So she did a lot of the preliminary work for these encapsulated plasmids, transfecting them into livers in a mouse, and then translates that into the canine system. And then the hope is to take the results from the canine system and translate them to people. But usually, yeah, people rely on preliminary work done in the mouse these days.
Other questions? Might be
I have
something.
I
Oh,
do have a question.
good.
So I just wanted to ask Brian just one question about the like usually like with the canine. So whenever you, for example, you treated like a dog, and what happened next? Basically like the fate of the dog. What what do you usually done with the dog after they are done treated? Like do you give them out for adoption, or do you like, keep them at the hatch, or?
That pretty much depends on what the experiment requires. Um, Quite a few of the dogs, yeah, are adopted after they're done serving their time here, and yeah, quite a few of them are adopted, and they go elsewhere and just live out their lives doing dog things. Occasionally, though, yeah, you know, euthanasia is an endpoint in some of these experiments. You need to collect tissues, and you need to make measurements that are experimentally necessary. So it really depends on the individual experiment. But yeah, quite a few of these dogs go elsewhere. They're adapted out to loving homes.
Okay, thanks.
Any other questions?
Yeah, I guess Kenny, I'm gonna list a little bit back to your question that Diane you'd had about sort of power, statistical power and number, and you mentioned that you generally just, it's a noticeable effect, or, I guess, I'm get stuck in my world where I'm like, oh yeah, a 30% increase, which is not so obvious all the time, so what drives how is it a difficult decision to scale up, that we need to scale up, that we need more animals into this, or how do you?
Well, the statistics do kind of dictate how many animals you will request and do the experiment on. So yeah, power calculations are a big part of it.
Mm-hmm.
But yes, that's why sometimes it's not really feasible, because sometimes they can be pricey if you start adding lots of animals. So you sometimes, yeah, like you have to make that kind of trade-off. So you need to have at least the numbers of animals that can give you the power that you need statistically, but like try not to have.
I have one more little question that might sound silly. I'm in a nutrition world. I'm in a nutrition department, and the beauty of rodents is we have these wonderful purified diets that we can highly control. Do those exist for canine models?
There is a standard lab chow that we give the dogs, but actually, like when they're feeling a little under the weather and they kind of have lots of appetite, you feed them like a whole variety of things, like chicken or some wet food. So there's not really like highly defined diets that I know of for dogs, but I'm sure that's something we can look into. Yeah, it seems like in nutrition you would definitely need that. I understand, but I don't know that those exist for dogs. I imagine they might.
Okay, if there aren't any other questions, we'll thank Brian again for his presentation. I think I need to go read a couple of canine papers to figure out how they're doing their power calculations and how many animals they use and things like that, so that I can see a graph of variability on things, at least for myself, to try and understand how you achieve statistical significance in the dog model. Thank you so much for your presentation, and thanks everybody for your participation. If you found this useful or you want to know more, I highly encourage you to, you can rewatch this, you can reach out to Brian. If you need to look for the website, you can go on that CCEH.io website, and there will be a link there in the services or the centers where you can get to the Canine Center at the Fred Hutch. And with that, we'll close out the presentation. Thanks again, Brian.
Thank you, Diane.
All right, take care.
All right, bye.